different populations evolve in different ways and at different rates. The evolution of populations can be thought of in the same way as the tortoise and the hare. Is faster or slower better? Who is the winner? Evolution is not a progressive process with a clear winner and loser, but an individual's fitness can still be measured as a marker of evolutionary success determined by natural selection. Let's dive deeper.
An adaptive landscape, as proposed by Sewell Wright, relates genotypes and fitness. It is described using topographical ideas, for example, genetic epistasis. The idea that one single gene's effect can be caused by the presence of other genes can lead to a rugged landscape, a multi-peaked evolutional curve. Depending on if the population is unstructured or structured, the metaphorical hare or tortoise, there may be benefits to both a rugged and smooth landscape.
An unstructured population in a rugged landscape has selective access to many or all of these peaks, but quickly approaches only one, so it may not necessarily reach the highest or most fit peak.
The spatially structured population is one whose selected traits and mutations may take longer to reach the whole population. Therefore, the furthest parts of the population are able to act almost completely on their own, allowing the population as a whole to experiment with multiple different peaks on a rugged landscape until the highest is found. Eventually, the entire population will reach the genotype found at the highest peak.
In other words, spatially structured populations take more time but evaluate all of the possible peaks in order to find the highest one, while unstructured populations gravitate to the first peak they find. Thus, the tortoise wins the race to the highest average fitness.
Adaptive landscapes with only one peak, otherwise known as smooth landscapes, do not have genetic epistasis, as every possible path ends up in the same place. Here, the unstructured population, the hare, beats the spatially structured population, the tortoise, to the top. In this case, speed is more important than searching all possible paths.
In May 2015, researchers at Michigan State University used this tortoise-hare idea in order to evaluate whether or not Escherichia E. coli evolved on a rugged or smooth landscape. They set apart two populations of bacteria, each with 96 subpopulations in different wells.
In the first population, known as the restricted population, migration was only allowed between the subpopulations directly next to one another.
In the second population, migration was allowed between all subpopulations.
Migration was simulated between the subpopulations by randomly exchanging bacteria of one subpopulation with the bacteria of another. In total, 36 migrations took place.
If the E. coli evolved on a smooth landscape, the unrestricted population would be the most evolved by the end of the experiment. If they evolved on a rugged landscape, then it would be the restricted population. The fitness of the bacteria at the end of the migration period was measured by examining the number of mutations between the two populations and their common ancestor. They found that the restricted population accumulated more mutations than the unrestricted population, making them more genetically different from their common ancestor and thereby covering more evolutionary distance. In a more theoretical context, researchers modeled the growth of E. coli bacteria, depicting the results of varying both the structure of the population and the ruggedness of the environment where they lived. Researchers ran these simulations of both restricted and unrestricted metapopulations that consisted of 96 populations each and found an outcome similar to that produced by the actual bacteria. If the researchers had found that the unrestricted population was more fit than the restricted population by the end of the model, then they could have concluded that E. coli evolves on a smooth landscape. However, because their results supported the idea that the restricted population was more fit at the end of the experiment, they could conclude that the E. coli population evolved on a rugged landscape, and the tortoise beats the hare yet again.
So why is this important? This research is useful in predicting evolutionary patterns based off of environmental constraints. For example, if we can pinpoint the moment where disease-causing bacteria such as tuberculosis becomes resistant to an antibiotic, we can release new antibiotics before they reach that evolutionary peak and prevent widespread population resistance. For example, one of the difficult parts of treating bacterial diseases is the necessity of releasing new antibiotics when the bacteria become resistant to the old drugs.